Okay, so up next we have Haoyun Lei from Carnegie Mellon, who will be talking about deconvolution of tumor copy number data using bulk and single-cell sequencing data.
Hello everyone, I'm very glad to present my ongoing work here, and I hope you guys will enjoy the talk, and I will appreciating suggestions and advices. As we all know, tumor heterogeneity is very important to understand the tumor evolution. The famous canonical evolution models illustrate that tumor can be arised from a single mutated cell and accumulate additional mutations as it progresses. And these changes give rise to additional subpopulations of tumor, and each of these subpopulations have the ability to divide and mutate further. When the tumor was treated by drug, although some tumor population would bottleneck due to the treatment, some drug-resistant tumor will survive and divide and then grow into a new tumor according to the model. So in order to develop some new novel drug combinations, we have to have more deep understanding about the tumor heterogeneity. There's two important methods to get the tumor heterogeneity. One is bulk tumor sequencing, and this technology can give us the average mutant allele frequency and the average allele copy number of gene in the tumor, and with some statistical method, the average canonical frequency can be inferred. Our previous work have developed a geometric model to express the relationship between the bulk tumor and the single cell Here is the example. Here is a very simple example for the model. For example, we have possible B and C and F, and the two data points B1 and B2 represent the tumor sample coming from the three mixture components C1 and C2, C2 and C3. We assume that the data are just assessed on two genes, G1 and G2. So the matrix B provides the coordinate of the mixture sample B1, B2 in the term of the copy number of G1 and G2. And the matrix F describes how B1 and B2 are generated from C. For example, the first row of F indicated that B1 is a mixture sample of equal part of C1 and C2. And this model can be expressed as a matrix dot multiplication with some constraint, which is the basis of our work. Based on the model, we can translate the biological problem in a mathematical way, and we can use a matrix to represent the average copy number of genes in tumor, in bulk tumor, and we would like to decompose the bulk tumor matrix into a cell component matrix that represents the exact copy number of each gene in each cell component, and the corresponding weight matrix that represents the cell amount of each cell in each tumor. One of our previous work developed an efficient gradient optimization method to optimize the geometric mixing for the measurement of tumor DNA copy number. This model can yield a very fast and accurate tumor state, even with very low and outlier data. But this model is based on the uniform distribution, which is not very right in the bulk tumor sequencing data. And our later work developed some exponential complex mixture model, and with some common pattern inferred from the genomic data, this model can resolve the discrete tumor subgroups, and then can accurately infer clonal heterogeneity from non-uniform bulk sequencing data. But this model requirement to better capture the real source of the genomic data. Since bulk sequencing is not good enough to infer tumor heterogeneity, the complementary single-cell sequencing data have been developed very fast this year. This technology can genotype individual cells, and it is the most objective and also direct inference of the gonadal genotypes, but it is too slow and noisy and very expensive for the large-scale data set. Since bulk sequencing and single-cell sequencing has some advantages and disadvantages, and some previous work have shown the possibility in combining them together to get a better inference about the cell subtype inside the tumor. There is one work last year that develops a very develop a novel object function that combines the train score infer from the bulk tumor and infer from the single cell data, and they show that the method outperform others that only use single cell data only. In our current work, we would like to get the gene copy number inside in different cell type inside the tumor using tumor and the single cell data together. Using the geometric model, we can pose the decomposition problem as we have bulk data B with size as the input with size M and N. Each row is the gene copy number in each tumor samples, and each column is the tumor samples. And we would like to get the output of a matrix of cell components C with size M by K, and M stands for the gene copy number, and K is the number of cell components, and corresponding with matrix F. The mathematical formula is like we would like to optimize the C and F, trying to minimize the Euclidean distance between B and CF with such constraints that each element in C and F should be non-negative, and each element in F is normally distributed, and each kernel of F should sum up to 1, and each element in C should be integer. The original optimization is non-convex, and it's hard to find the global minimum, but we can fix F, we can fix C or F, and iteratively to optimize F and C, so we can get a good global minimum, local minimum. According to the non-negative, the work of the non-negative matrix factorization, the Euclidean distance between B and CF is non-increased under the multiplicative updated rule. The original NMF algorithm can work for this problem. However, we are trying to infer the copy number of the gene, which is undeterministic, and the original NMF algorithm starts from random initialization, so the average accuracy is very low. So from the biological aspect, genes are dominantly deployed. With this knowledge, we gradually set the entrance of the initialization to deploy it, and by doing this, we find the average accuracy significantly improved. And for this idea, in another data set, in another set of experiments, we replace the random initialization with some real single-cell data, and we find that the average accuracy improved further. So although using some real single-cell data as the initialization can work much better, there's still room to increase the accuracy. We found a way to modify the object function by adding some auxiliary penalty. At this point, the same assumption is that the cell component we want to infer should be very similar as the cell component we use to simulate the tumor. So we in the penalty we included some included some original single cell data we use to simulate the tumor as a part of the matrix, and we try to compare the Euclidean distance between the two matrices we want to two matrices. In another direction, we use the train cost to compare the cell component we want to infer and the cell component we use to simulate the tumor. For example, we have the cycle here represent for the single cell data we don't know, and we try to infer another 5 dots field 5 field dots represent the single cell data we don't know, and we keep the cost of the tree minima. Our improved matrix decomposition method for the bulk tumor deconvolution can work well on very large data sets, large scale data sets, but it's kind of unrealistic to have so many tumor samples in real world. To resolve this problem, we develop another way to simulate the tumor samples in two steps. In the first step, we use a geometric mixed model to simulate the tumor samples from the cell component, and then in the second step, we still using the geometric mixture model, combine the tumor samples with the cell component to simulate a high-level mixture matrix. By doing this, we are able to use the existing matrix decomposition method to decompose the high-level mixture matrix. After introducing all the methods, I would like to show some representative results. We are working on semi-simulated data for now. The figure shows that the hierarchy structure of the tumor. Each tumor contains different regions, and each region contains a lot of single cells, and each single cell sequencing file contains a lot of information about the gene. With the help of our collaborator, we be able to get access to the single cell sequencing file of Glioblastoma, which is very common brain cancer, and the single cell file have the similar structure shown here, and we only extracted copy number from the file to compose the cell components. And by using the geometric mixture model, we simulated the matrix F such that each element is normally distributed, and each element is non-negative, and each corner of the F is normal to 1. And then we can simulate the tumor samples as to the desired number we want. This figure shows one instance of the pure bulk data decomposition. The left figure means the original copy number of genes in the cell components we use to simulate the tumor, and the center figure shows the stands for the fraction copy number we inferred from the pure NMF algorithm, and the red figure is that we run the fraction copy number to the nearest integer. And in this particular good examples, almost all the genes copy number are correct, which means the pure MMF algorithm sometimes works for this problem. This result shows the statistical analysis on the decomposition method with different modification of the matrix first we compare the pure MF decomposition with some deployed in the initialization. We can find that with just random initialization, the NMF algorithms have very low accuracy, while when we gradually introduce some deployed into the initialization, the average accuracy increase significantly. And then the right three figures is that we replace the random initialization with some real single cell data, and and we find the average inaccuracy continue to decrease. So we can increase the average accuracy from 5% to 9% in this when we introduce some real single cell data into the initialization. And this result comes from the decomposition with auxiliary penalty. On the top, we can see that when we increase the regularization parameter to 0.2, the average accuracy almost reaches 1. And in that one instance, we can find that only very few number of genes, the copy number are not right. And at the bottom, we compare the L2 and L1 distance with distance objective functions with matrix comparison and train cost. The matrix comparison method can return very high accuracy, but it requires a large number of tumor samples. And the train cost method can return less high accuracy, but it does not require too many simulated tumor samples. Finally, we are able to show that with the two-step simulation, we can work on a very small number of tumor samples, although we have lost some accuracy compared to the experiment in the the experiment that were done with very large number of tumor samples. Here is some conclusion for the current work. We have shown that with limited single-cell sequence data, we can infer a cell component with a very high accurate copy number from simulated bulk tumor. And the basic approach can achieve very high accuracies, but require on Heuristic a large number of tumor samples. And we develop a way to deal with it, although we have lost some accuracy. Our future work may lie in two main directions. First of all, we have been developing some more advanced evolutionary model based on the phylogenetic tree. As I mentioned in previous slides, the tree construction can work in smaller number of tumor samples, but only with very small number of genes. And we are trying to make it that works with a large number of genes. Second, we would like to integrate some other class of genomic data, such as the breakpoint in chromosomes that were used by our former lab members who gave a talk yesterday. And finally, I would like to thank my advisor, Dr. Schwartz, and other lab members, and I also would like to thank our collaborators. Thanks all for listening. Thanks.
Do you have any questions?
Hi. So I'm from a more mathematical biology background, so maybe my question is a bit ignorant. But if you have you have data about single cell, is there a chance that the variations you observe are simply noises rather than any meaningful phenotypic differences? If that were true, it would mean that the coarse-grained approach is actually better, despite having a lower resolution. What do you think?
you're talking about one single cell data or not?
Well, I'm saying, because you're saying one single cell data is more accurate because of the high resolution.
Yeah.
But if you have data about one cell only, chances are the variations are simply noises.
Right.
And you're not really interested in those fluctuations. Is there this possibility?
I think so, but yes. But I mean, because we have a lot of single cell data, but I mean,
yeah. So in that case, maybe one way to do it, that from my point of view as an outsider, is to use your coarse-grained average data to build an average model, and your single cell data, they are just the error function. Would you say that is a productive way to do things?
actually, I'm not sure. Sorry.
I think we maybe take this conversation offline. But thanks for the question, and let's thank the speaker again. Thank you.